Welcome to 13 Universal with Shedro. This is A-level CIA biology. Okay. And guess what? All topics, paper 4, series 9. We have done paper 1, 2, 3, 4, 5. Okay. 6, 7, 8. This is 9. Thank you for following us. Make sure you share the video. Make sure you recommend the video. So don't forget that these questions are from CIE. So let's start. Get your writing materials. Remember, what we do here is that we apply concept. We try to apply concept. Okay. So let's solve questions. Do not check the math scheme. Do not check the math scheme. All right. Make sure you follow through. So Fig 7.1 is a diagram representing a synapse between a chemoreceptor and a cell from human test board and a dendrite of a sensory neuron. Okay, so this is chemoreceptor, is a receptor, so it's in the tongue. All right, so this helps to dictate all those sweet taste of yogurt, cheese, that ice cream, pizza. So this is in your tongue, and chocolate. So, and of course this dictates the stimulus and then generates impulse and then transmits it to the sensory neuron. That's the dendrite of sensory neuron. Okay, so remember that once impulse arrives, and it's a strong impulse, all right, so remember that action potential will be initiated here, right? So the membrane potential will change, and then neurotransmitters will now carry the impulse. All right, neurotransmitters released by this, all right, goes to this. Okay, and then initiates action potential here. Did you get? All right, so let's go to the question. Remember that whatever happens here affects what happens here. All right, so you see, in an experiment, different concentrations of sodium chloride were applied to the microvilli of the chemoreceptor. All right, chemoreceptor cell. The membrane potential of the chemoreceptor cell and the membrane potential of the dendrite of sensory neuron were recorded for each concentration. So look at this. Which one is the independent variable here? Let me show you something, because this is an experiment. Okay, so we have different concentration. Okay, concentrations of sodium chloride. We apply to the, and then we now have membrane potential of the receptor and whatever, okay, measured, right? Okay, so which of them in this experiment is independent variable? Concentration, because as you read you have to think together. Concentration of sodium chloride or membrane potential, which one do you choose, guys? Of course, concentration of sodium chloride, because we are changing this, right, and then we're measuring that. All right, let's go on. The resting potential of this chemoreceptor cell is minus 50 millivolt. That's minus 50 millivolt. And the resting potential of the dendrite of this sensory neuron is minus 70 millivolt. This. So what is resting potential, guys? Resting potential is the potential, membrane potential of a receptor or a neuron when it is unstimulated. Okay, when it is not transmitting any impulse, it's unstimulated, it's resting, actually, literally. The results are shown. So let's go. Remember that what happens here determines what happens here. So this is the result. So explain the results shown in table 7.1. Yeah, pause this video and try it. Explain the results shown in table 7.1. Okay, so explanation demands that you're going to tell us the meaning of this result. If they are said describe means show us the trend, okay, and the relationship. Explain means use biological concept to explain this, to interpret this. What is the meaning of this? So they're asking, what is the meaning of this? What is the meaning of this data? Okay, so first of all, this is the independent variable concentration of sodium chloride solution. All right, don't forget to share this video, share with your friends, and we're going to start topicals, right, very soon. Now, can you see that at 0.1 gram per dm cube, the membrane potential of chemoreceptor is still minus 50, which means it is not depolarized. And because it's not depolarized, action potential is not generated. Therefore, dendrite of sensory neuron is also not depolarized because information flows, impulse moves from chemoreceptor to dendrite. This is not depolarized. This is not also not depolarized. But look at this. I'm explaining this. As the concentration increases from 0.1 to 1.0 and then 10.0 gram per dm cube, what is happening here? You can see that the membrane potential, all right, changes from negative minus 50 to, all right, plus 30. So there's depolarization occur, and this has been depolarized, okay, and therefore action potential is generated. This is a peak action potential. So because action potential is generated here, right, you can see that action potential is also generated here. Why? Because this is explained. Why? The reason is because action potential is generated in the chemoreceptor. All right, that action potential is generated because the membrane potential, of course, the membrane potential is greater than the threshold potential. The threshold potential is usually minus 55 because it's greater than threshold potential. Remember that the arrival of stimulus, all right, causes the membrane to become depolarized. So because change in membrane potential. So because it is depolarized, if that change in membrane potential is greater than the threshold potential, they have action potential initiated, which is now called, of course, that that will cause neurotransmitter, okay, to be released, okay, into the synaptic cleft, and then that causes membrane potential, action potential to be initiated in the dendrite because information stimulus move from here to here. All right, okay, let's not waste further time. So you see at 0.1, okay, it's still depolarized resting potential. Nothing is released, resting potential the same. Because of that, the dendrite is not depolarized either. But at this and that is depolarized, so it releases neurotransmitter. Therefore, depolarization action potential occurs in the sensory neuron. Okay, so you make mention of the fact that membrane potential, change in membrane potential in the chemoreceptor is generator potential, and of course is greater than threshold potential, and that's why action potential is initiated. Okay, all right, let's go to the next question, guys. Describe differences in the structure and function between sensory neuron and motor neuron, right? So you know how it is. Pause this video and try to answer this question. All right, so what am I drawing? What do you think I'm drawing? What am I drawing? Okay, yeah, do you remember this? But meanwhile, you must have paused this video, okay, to work on your own. Now you are back. Join me, let's crack this. So sorry for this, but I'm just sketching it, the structure. So yeah, they're all unmelanated, okay. Yeah, now what is that? Don't forget this goes like this, and then we'll have this, okay, and maybe that something like that. Okay, so we'll have this. So what is this I'm drawing? This is dendrite, okay, dendrite, dendrite. Okay, so dendrite, right. Now let's look at this. Is what neuron is this? Which one is motor? Which one is sensory? This is a sensory neuron. This is motor neuron. Okay, so let's look at differences in their structure. If you remember, so you see this is cell body. That is where nucleus is found and other organelles are found. All right, this is okay, cell body here. The cell body of sensory neuron is between the axon and the dendrite. Now the axon, don't forget, is the cytoplasmic process that carries impulse towards the cell body, while axon carries it away to the next neuron. Okay, so you can see, guys, that this is in between axon and dendron, while the cell body of motor neuron is at the end of the neuron. Okay, it's at the end of the neuron. Okay, so what is the next difference? You can see that motor neuron here doesn't have dendrite. Motor neuron or sensory neuron, sorry, not motor neuron, the cell body, sorry, the cell body of sensory neuron, this is the cell body of sensory neuron, has no cell dendrite. Cell body has no dendrite, while the cell body of motor neuron, this has dendrite. Okay, yeah, very important. And that thing I wanted to note is that you see that this has, all right, axon and dendron. This is dendron. Okay, all right, and of course but this has, you can see axon here. The axon here is longer. It has long axon here. Okay, but this has axon and dendron. Short axon. This is long axon. Right, now of course this is found in the spine, ganglion or spinal nerve, while the soma here or cell body is found in CNS. Okay, so the question is, what is their function? The function of sensory neuron is that it carries impulse from the receptors to the central nervous system. That's for sensory. While motor neuron carries impulse from the central nervous system to where? To the effectors. Okay, so let's see what it said here. All right, see that? Check it out. Cell body between dendron, cell body between dendron and axon. Okay, or in ganglion. Cell body is at the end, so it has dendron and axon. This has long, this one has long axon. Okay, no dendrites from cell body, no dendrites here, but these dendrites are found here. Okay, carries impulse from receptor to CNS or intermediate. It carries impulse from CNS intermediate to effector. Okay, let's look at this. Describe the functions of the internal membrane of the chloroplast. Now, get ready. This is about 7 marks question. I like you to solve this question. Okay, describe the functions of, anytime they say describe, all right, they're asking to give the characteristics and their role. Okay, give the characteristics, the major features here, give the characteristics, all right, of the internal membranes of the chloroplast. Permit me to quickly pause this video and start solving it, guys. Make sure you are solving this. All right, so I'm solving my own. So what do you think is this? What is this? Oh, I didn't get it well. Let me try again. There's no harm in trying. Let me try to sketch something. Oh, did I get it? Did I get it? Okay, didn't get it properly. Okay, now of course, what am I drawing, guys? Come with me. I'm drawing, oh, shouldn't touch. I'm drawing something that bakes bread for us. This thing fries our chicken, makes our ice cream, and then gets our pizza ready. Okay, now this is just a sketch of an organelle. What's the name of that organelle, right? Okay, that is chloroplast. You can see this is a folded membrane, that's thylakoid, stack of thylakoid on one another, okay, to form granule. Granule is a stack of thylakoid, right? This is another stack of thylakoid, right? Stack of thylakoid. You can see stack of thylakoid. That's another granule. A stack is granule. Folded membrane is thylakoid, right? So they say you should describe the structure. Now don't forget that this is joined to this by another membrane called lamella. So lamella connects the granule. Okay, guys, the first structure here I'll mention is that chloroplast has internal membrane system. Okay, that internal membrane system is called grana. Okay, grana. Can you see grana? That internal membrane system. Grana consists of a stack um, of thylakoid granule. This is a granule, that's another granule, that is another granule. Okay, so now that provides large surface area for light absorption. That's the first thing I'm describing. It has large surface area for light absorption. Okay, now that's very important. The next I'm going to mention, let's go in. So I'll now mention that, of course, the grana consists of a stack of folded membrane called thylakoid. Now this is thylakoid, is folded membrane. See it is a membrane. That means it has phospholipid bilayer. If you zoom in, you're going to have this phospholipid bilayer. That is a membrane. Okay, so we call it thylakoid. Now thylakoid membrane is a site for light dependent stage of photosynthesis. So this guy is a site of light dependent stage. That's thylakoid. So what feature makes it a site? Now this is the membrane here. Okay, that's, if you zoom in, this is the lumen. Why is it a site for light dependent stage? Number one, because it consists of photosynthetic pigment. So this photosynthetic pigment are arranged in light harvesting clusters called what? Called photosystems. So photosystems are called LHC, light harvesting clusters. All right. So if you look at it, so this photosystem consists of the primary pigment, which is usually at the base of the, that's the reaction center. Can you see? This is like antenna. You can see the antenna I'm drawing. This antenna complex is the accessory pigment. So we have primary pigment and accessory pigment. Okay, so they have different roles. Okay, the primary pigment absorb wavelength that, the accessory pigment, all right, absorb wavelength that primary pigment cannot absorb, and then transfer the energy to the primary. See, so it absorb wavelength that primary cannot absorb, and then pass on the energy of the wavelength. Okay, so it has two roles: absorb wavelength that primary pigment cannot absorb, and then pass it on. So that passes it on. All right, it passes on to the primary pigment. Don't forget that. Do not forget that. And then what happens is that the primary pigment, okay, if you check the primary pigment, what is the function of primary pigment? What is the function of primary pigment? So the primary pigment absorb wavelength. All right, it can absorb wavelength. All right, but the major function is that it is the reaction center. So it is where photo activation or photo excitation of surface electron occurs. So the primary pigment is where electrons, all right, are excited. They move from lower energy level to high energy level as they absorb light or photons. So these surface electrons, they jump from, right, one energy level to another. So that's photo excitation. I'm showing you photo excitation, right? Okay, so when they reach the highest energy level here, so the electron is emitted. So you see the electron is emitted, that's photo emission. Okay, so it leaves the surface and then enters into electron transport chain. Okay, so these electrons start moving down from one electron carrier down energy level. So this is from one carrier 1 to another carrier 2. So it's moving down energy level. Okay, all right, so maybe carrier 4 here, but as moving down energy level, is losing energy. Okay, so the final electron acceptor here is NAD, okay, P. All right, so NADP combines with electron and proton. It combines with electron and proton. Okay, all right, to form NADPH. Remember? Okay, that's what is happening here. NADPH. All right, NADPH. Okay, NADPH. So I'm just trying to explain this quickly. All right, now look at this, guys. Let's get into the game. Because the energy that is released by the electron is used to pump proton into intermembrane, into thalocode membrane. So from stroma, the energy released here is used to pump proton from stroma to the thalocode lumen. Stroma is moved from here to here, and then proton gradient is set up between the thalocode lumen and stroma. So proton move out back through ATP synthase. As it moves down concentration gradient, protons causes ATP synthase to join ADP and inorganic phosphate, and that process is called chemiosmosis. All right, let's come back here. Now, describe the functions of the internet. So the first one to mention is that this has large surface area, granular, okay, made up of thalocode membrane. Thalocode membrane is a site of light-dependent state because it contains photosystems. Okay, the first thing has photosynthetic pigment, right, which absorb different light wavelengths. Okay, so they are arranged in clusters called photosystem. The photosystem is called light harvesting clusters. Look at them here. So there are two types. We have the primary pigment and the accessory. Accessory pigment form the antenna complex, while the primary pigment forms the reaction center. So accessory pigment examples include chlorophyll B, xanthophyls, and carotenoid. All right, the function is that they absorb wavelength that primary pigment does not absorb and then pass the energy to the primary pigment. Then primary pigment absorb wavelength too. The primary pigment, all right, the primary pigment is chlorophyll A. All right, it is a reaction center. That is, it is where photoexcitation of electrons goes on. Can you see electrons jump from one level to another until they're emitted? All right, so this is photoexcitation, this is photoemission, right? Now this membrane also consists of electron carriers, which allow the transfer of electron in the electron transport chain. Okay, so when electrons are moving down, all right, so the energy is used to set up concentration gradient. Okay, so protons concentration gradient set up here. So protons move out and then cause ATP to be synthesized by passing through another protein called ATPase embedded here. So this has photosynthetic pigment photosystem. It has electron carriers. It has ATPase, which synthesizes ATP. All right, so guys, yeah, we have dissected it. So let's look at what mass scheme says. See, photosynthetic name pigment, all right, accessory pigment to absorb light energy, reference photosystem, antenna complex, reaction center. Okay, so guys, so you see the sequence. Each of these parts, accessory pigment, they absorb, okay, light energy. All right, so photosystems, antenna complex, reaction center, all of them harvesting structures, photoactivation of electron. I think we mentioned that electron move along the electron transport chain because we have electron carriers embedded in this thalocode. So that's called photophosphorylation. Okay, all right, and also we have photolysis of water, which occurs also in the, all right, it occurs in the photosystems, all right. So, and you can see that, so because it contains an enzyme called oxygen evolving complex, so thalocode and stack to rather give large surface area, space, or lumen. Yeah, this lumen, all right, creates proton gradient. Okay, so ATP synthase also make ATP. So all of these are contained in this, and they have different functions. Okay, all right, that's 7 marks. All right, so this is another question. Rubisco activase R is an enzyme that has an effect on the activity of Rubisco. An investigation was carried out on effect of Rubisco activate on the activity of Rubisco. Solution of Rubisco and RBP were added to two tubes A and B. RA was added to tube A. Both tubes were incubated at 6 minutes, 25 degrees Celsius for 6 minutes. The activity of Rubisco was measured every 30 seconds. You can see that. All conditions were kept the same except for the addition of RA to tube A. So RA was added to tube A, but it wasn't added to tube B. You can see. Okay. All right. Every condition constant. Let's see. Describe the resorption 8.1 and suggest an explanation for the effect of RA on the activity. So if you describe, there are two questions: describe and then explain. So it's explanation. So in description, you show trend. You can see that the Rubisco activity in tube A increases over time, while that of B remains constant. Okay, so you can quote data here. So you can see that it increases from, because from here to here is okay, 1 divided by 5, that's 0.2. So you can see this is about 0.5 minutes, right? It increases at 0.5 minutes. This is about this is 0.4, okay, about 3 point, I think 3.8 or thereabout. Then it increases by the time it is 6 minutes, it has increased to about 11.6, okay, activity arbitrary unit. So quote the data. But the next thing is explain. Make sure you are solving this question. You're pausing the video and solving it. Okay. Explain. What is the explanation? The explanation is that, all right, this Rubisco activase that was added to tube A has effect on the Rubisco. What effect? Okay. Now, what effect do all these molecules that that activate other molecules do? So either that it causes it to change the shape of the active site so that it binds more with the substrate RBP to form more ES complex. Okay, all right, and then or that it helps it to remove more of the product from the active site so that it can bind more. Okay, it can bind more to the substrate. Okay, so that's it here. So you see the description part, so you have to state the trend and quote data. Now this is explanation part. Rubisco activates okay by changing the activity. So why does it increase? What is the effect of Rubisco activates? Okay, of course activates the, so it activates it, okay, by changing the active site, all right, of Rubisco, or by enabling it to bind more readily. Yes, enables it to bind more readily so that more enzyme substrate complex form, or enables product to leave. Okay, it's acting as a cofactor. Okay, all right guys, let's look at this. Fig 9.1 is a diagram of a relaxed sarcomere in straighted muscle. You can see that. Yeah, that's sarcomere, right? So from here to here, the sarcomere. A band, okay, mixture of thick and thin. I band only thin. H band wasn't given here, which is here from here, thick. Okay. So what's the question? On Fig 9.1, use label lines and letters to label an actin filament with letter P, myosin filament with R. So pause the video and try it. Okay, actin is a thin filament. So label here with P. This is a thin filament. This one here. So this is P. And the thick one is R, myosin. That's myosin, okay? So that's it. All right, so pointing to thin filament, okay, then pointing to thick filament. 2 marks, no time. Fig 9.1 is a diagram of a relaxed sarcomere. Okay, the same question, look at it. Yeah, they said state what happens to A band. This is band, not base, and the I band when sarcomere shortens. You see, when sarcomere shortens, see, this thin filament will move this way and this will move this way because, you know, because the myosin is pulling it. It pulls actin. Actin is being pulled in this direction here, okay, and also it's being pulled in this direction here. All right, it's being pulled in this direction here. Where is it? It's pulled in this direction here. Okay. So what happens to the A band? This is A band. A band is from here to here. So this is not moving. It's not moving. So what is moving is this. All right. So A band remains the same. It's only this one that's been pulled this way. What of I band? I band is the part, right, that consists of only the thin filament. So when you pull the thin filament, this I band will shorten. Okay, if you pull it like it will shorten. So this shortens or narrows. I band shortens or narrows, while A band remains the same. The same way sarcomere also, because this is going to come this way, this is going to come, sarcomere shortens. But A band the same, while I band narrows. Okay, all right. 2 marks. No time. The plant's Toxosphaera produces toxin Caie, which causes muscle paralysis in mammals. The toxin acts by binding to receptors on the cell surface membrane sarcolemma of muscle cells at neuromuscular junction. Suggests how binding of Caie to receptor may cause muscle paralysis. Guys, you remember that if you have done this, pause this video and try it. Okay, pause the video and try this. Okay, try to describe what is happening here. All right. Okay, so all right, pause this video and try to describe it. Okay, you can see what is happening here. Okay, so remember, there's a suggest how binding of, so when Caie, you see, this is neurotransmitters. They are released and they bind to their receptors here and open sodium ion channel, so sodium ions can enter and cause the polarization here. So this is presynaptic neuron, which is motor neuron. This is the muscle, all right, muscle fiber. This membrane is sarcolemma, right? That's acting as postsynaptic membrane. Okay, this is synaptic cleft. Okay, so now this is just how binding of Caie to receptors, right? So when they bind to receptor, they block the neurotransmitter. When Caie bind to receptors of neurotransmitters, they block it. And once they block neurotransmitters from binding to their own receptors, then sodium ion channels do not open. So sodium ions, or no sodium ion or few sodium ion enter the muscle fiber. Okay, no or less action potential generated, or no depolarization occurs, right? And if it does not occur, so calcium ions do not move out of sarcoplasmic reticulum. They don't move out, and therefore a few of them move out, or okay, and that will lead to less troponin, because calcium ions bind to troponin. So if you have few or no calcium ions that leave the sarcoplasmic reticulum, that means few or no calcium ions bind to the troponin, and troponin does not change its shape, and then myosin binding site is not uncovered, and then my myosin head doesn't bind, so there's no cross bridge formed between myosin. All right, so remember, the prerequisite to solving questions that you must have done the topic and understood it. Okay, NE4 complete with acetylcholine or neurotransmitter. So that Caie blocks the receptor. So sodium ion channels here on the postsynaptic membrane does not open. So no or few sodium ions enter the muscle. This is muscle fiber, okay? And of course, no or less depolarization of the sarcolemma. Okay, not the, okay. Number 4, no few action potential generated, does not spread across muscle, and calcium ion voltage gated channel do not open the sarcoplasmic reticulum. So no of your calcium ions bind to troponin, no exposure binding site. Okay, you know how it works. So 4 marks, any of the 4. The plant Toxosphaera produces the toxin Caie, which can cause muscle paralysis in mammals. The toxin acts by binding to receptors on the cell surface membrane or sarcolemma. Okay, yeah, I think we have done that before. This suggests why action potential, okay, why the action potential of Caie may lead to death. Remember that when it blocks, there's a problem here. If Caie blocks the receptor, maybe it has similar shape to the receptor, similar shape, all right, to the neurotransmitter acetylcholine, and maybe it's complementary to the receptor of this neurotransmitter. So it binds to the receptor of neurotransmitter and blocks it. That means there's not going to be transmission of impulse, right? This suggests why the action of Caie may lead to the death of mammal. So guys, think about all the muscles that a mammal uses. For example, you can start with the skeletal muscle, all right? So that means if it leads to paralysis, this mammal will not be able to escape from predator, it can run. So it's going to be eaten, okay? That's one of the things that may happen, lead to the death, or is not able to feed, okay? All right, because it cannot pursue the prey. It doesn't feed or go to grass to feed, so it's going to die. or if it is the smooth muscles, okay, that helps in breathing. The smooth muscle does not contract, okay, so there's no inhalation, no gas exchange takes place, no oxygen is taken up, and then they can die after a while, okay. Or the cardiac muscles of the heart do not contract, so there's no blood circulation and oxygen is not delivered, and the guy dies. The guy is in trouble. This cruel is dangerous. Poisons are dangerous. Can you see? Okay. Affect rib muscles. The last one is that the intercostal muscles or diaphragm, unable to breathe. Affect cardiac muscles, so stop blood circulation, heart failure or attack. Cannot move, so unable to escape from predator. Okay. Cannot eat, so starves to death. So this poison is the plant. Okay. When you see this plant, run for your life, guys. Run. The name is scary. Complete the passage by using the most appropriate scientific term. Look at it. Okay, let's now pause this video and complete it, guys. Pause the video and complete it. So biodiversity within an area can be assessed at different levels, including the species. Okay. See, biodiversity, okay, and ecological diversity. See it. Species diversity can be assessed by determining the number of different species and the relative abundance of different species in a given area. From this information, species diversity can be estimated using Simpson's index of diversity. Organisms with the same species can show more genetic diversity even though they share the same genes. This is because they have different combination of alleles. The greater the genetic diversity, the greater the ability of a species to adapt to a changing environment. Ecological diversity is a measure of the number and range of different ecosystem and habitat within an. So let's see. So that's it, guys. 6 marks, a lot of marks. The International Union for Conservation of Nature IUCN red list of threatened species is updated regularly. Table 10.1 shows the number of endangered animal species counted every 3 years between 2007 and 2019. You can see that, guys. That's it. So the first question: calculate the rate of increase in the number of endangered species between 2017 and 2019. Show your working. Give your answers to the nearest whole number. So rate of increase. First of all, find the increase. All right. In 2007, the number is 7,851. 2019 number is 14,234. So pause the video and solve this calculation. So let's find increase first. So increase is this minus this. Okay, 14,234 minus 7,851. So it has increased from year to year. So let's find increase. So get your calculator, punch 14,234 minus 7,800 and that's 7,851. Okay, so you see, when you point the calculator, it gives us 6,383. 6,000, that's the increase. 300 and 6,300 and what am I writing? Okay, let me write it well. So the increase is 6,383. Good. Right, that's the increase. But they say we should find the rate of increase. Rate includes time. Okay, now remember that it takes how many years from 2017 to 2007 to 2019. 19 minus 7 is 12 years. So 12 years. So rate is what is the number in 1 year. So you divide this by 12. So this by 12 years, right? Okay, so that divided by 12 gives you what? 531. Okay, 0.1. You answer to nearest whole number. So nearest whole number is then 532. I guess so. 32. Okay, yes, that's the rate. Right, let's say mass scheme. Okay, see it, minus it, and give it. Okay, that's it. So let's look at next question. More species of fish were listed as endangered in 2019 than species of mammals. So more species were endangered. So just reasons why more fish species than mammals were endangered. Okay, more fish. Pause the video and try it. Why do we have more endangered fish species than mammals? Probably, all right. There's more hunting. All right. So no laws stopping, fewer laws stopping regulating hunting of fish, okay, fish species, okay. Maybe another thing you can say, more pollution, right? People dumping plastics, nylon, which could harm fish, okay, right? Or it's difficult to enforce those laws against, right, overfishing, right? And then of course, because you can check for mammals too. Now that means that there are more laws for mammals, right? More laws regulating hunting of mammals. Okay, is there are more, okay, laws, and is more is being enforced. But don't forget also that maybe fish has more predators, predatory fish increase in number. Okay, less food for them because of competition. Look at it, guys. So overfishing on water is difficult to enforce the law. Fewer laws to protect the fish. There's trophy hunting people. Climate change, yes, increase. So that causes predatory fish to move into new area. Pollution, plastic, oil. So these are the reasons. Okay. So there's more conservation project for mammals. Okay. So look at it very well. Remember that when they ask you to suggest, they want you to think outside the box. So thank you for staying true. It was a long video, but make sure you follow up on the next series, topicals. We are going to topical paper 2, starting from photosynthesis all the way to gene tech. All right. This is schedule. See you. Right. Bye.